Hi all, the title for this assignment is The Emergence of Bovine Tuberculosis in African Buffaloes discovered in South Africa's Kruger National Park and the role of fruit bats and primates in the recent outbreak of Ebola virus in West Africa. Introduction. Emergence of wildlife infectious disease pose a significant threat to wild animals, domesticated animals, and human population. Infectious diseases affecting wildlife has recently caused a decline in their population, and they also become reservoirs or maintenance hosts and cause disease in domesticated animals and in human population. Expansion in human population is one of the important factors which has increased the emergence of wildlife diseases by causing shrinkage in wildlife habitat, which increases the population density, and subsequently these animals become susceptible to diseases through coming in close contact with other reservoir animals which could be wild or domesticated. Factors such as increase in public awareness and active surveillance of wildlife has increased in the reporting of infectious diseases that are currently affecting the wildlife population. Therefore, this presentation highlights two major diseases associated with wildlife. One, bovine tuberculosis, which is threatening the population of buffaloes and lions in Africa, and two, Ebola virus, which has close links with fruit bats and primates, and it has caused significant health issues in human population in recent years. Firstly, bovine tuberculosis and its history. Bovine tuberculosis is a chronic infectious disease which affects a wide range of mammals, including humans. The disease existed in Europe from a very long time, and it was spread by European settlers to other continents during colonization. And the disease was first discovered in South African cattle in the 1880s. Distribution of bovine tuberculosis. The map on this slide shows the current distribution of bovine tuberculosis globally. The disease occurs worldwide and affects cattle and other bovids, which is why it is named as a bovine tuberculosis. The disease has been eradicated in Australia and is currently present in marsupials in New Zealand, cervids in North America, swine in Europe, and in cattle and wildlife in Africa. Bovine tuberculosis is still widespread in Africa, parts of Asia, and in some Middle Eastern countries. Outbreak of bovine tuberculosis in South Africa. The disease was first diagnosed in African buffaloes in South Africa's Kruger National Park in 1990. It is believed that the buffaloes got infected by grazing with the domestic cattle around the park. These cattle, which may have already been infected, acted as reservoirs and transmitted the disease to these buffaloes. Epidemiology of bovine tuberculosis in Kruger National Park. Firstly, host and wildlife affected. The slide shows that cattle are the primary host of bovine tuberculosis, and it transmitted the disease to African buffaloes in Kruger National Park. After contracting the disease, the buffaloes became maintenance hosts and spread the disease to other buffaloes within the herd and to other wildlife animals such as lions, kudu, and warthog. It has also been reported that cheetah and leopard in Kruger National Park has also been infected with Bovine tuberculosis. Other wildlife which have been reported to be infected with bovine tuberculosis in other parts of the world include wild boar in Europe, brush-tailed possum in New Zealand, and white-tailed deer in North America. Pattern of disease spread in Kruger National Park. It is believed that the disease was first introduced in Kruger National Park between the 1960s and 1980s from domestic cattle, but discovered in buffaloes in 1990. Survey carried out at the park suggested that bovine tuberculosis has been initially introduced to buffalo from infected cattle grazing in areas surrounding the southeastern part of the park during the 1960s to 1980s. Since the southern part of the park had the highest disease prevalence in African buffaloes than the northern and the central areas. The map on this slide also shows how the disease is spreading from the southeastern part of the Kruger National Park towards the central and the northern region of the park. This could be due to the movement of the animals, especially the movement of the buffaloes from the southern part of the park towards the central and the northern part of the park, and apparently while they're moving they are spreading the disease to the other buffaloes in the park. Cause of bovine tuberculosis. Bovine tuberculosis is a zoonotic disease which is caused by bacteria known as Mycobacterium bovis. This bacteria is a gram-positive bacteria and it is the most important species in the Mycobacterium tuberculosis complex that can cause human and animal tuberculosis. Symptoms of bovine tuberculosis in buffaloes. Within 3 to 6 months of infection, affected buffaloes develop lesions in the lymph nodes of the head, tonsils, thorax, and lungs. Adult buffaloes will remain infected for 3 to 5 years before mortality. The disease also causes weight loss, animal becomes weak, have dry coughing, also have dull coat, have depression, and become lame. Buffalo herds affected by bovine tuberculosis normally have a very poor body condition which worsens in dry season, and they can easily become susceptible to predation by lions. Disease transmission in Kruger National Park. As described earlier, African buffaloes originally contracted bovine tuberculosis by being in close contact with the infected cattle grazing around the park. Once infected, then buffaloes become maintenance hosts and can transmit the disease within their herd to other buffaloes or to other wildlife animals. Buffaloes are social animals, and one herd of buffalo may contain about 50 to 500 animals, and they can directly spread the disease to other animals when they are in close contact. Some of the routes through which they can transmit the bacteria are: one, through respiratory routes, whereby the animal can inhale or ingest bacteria through aerosols or droplets through respiratory tracts. Secondly, the bacteria can be excreted in feces and urine of the infected animal and then consumed by another animal. Example, buffaloes live together and graze together, share habitats, and congregate together, which facilitates the spread of bacteria from infected animal to the healthy animal. The slide shows a picture where a herd of buffaloes are drinking near a water hole. This could be a effective way in which the buffaloes can manage to transmit the bacteria to another buffalo because they are all in close contact with each other. Bacteria also spreads through predation, especially when animals such as lions prey on the infected buffalo. They consume the infected material such as lesions, animal tissue, blood, and internal organs. The picture on the slide shows a couple of lions attacking a buffalo which could be infected with bovine tuberculosis. This route of transmission is becoming a major issue in Kruger National Park as other carnivores such as cheetah and leopards have been reported to have become infected with bovine tuberculosis. Survival of the bacteria. The bacteria causing bovine tuberculosis can survive for several months in the environment, especially in cold, dark, and in moist conditions, where the temperature can range from 12 to 24 degrees Celsius. The bacteria can survive for 18 to 332 days. In dry and moist condition, where the temperature is generally about 34 degrees Celsius, the bacteria can survive between 4 to 8 weeks. And during summer, when the temperature is generally higher than 34 degrees Celsius, the bacteria can survive for about 4 days. Impact of bovine tuberculosis in African buffaloes. Kruger National Park has approximately 27,000 buffalo population, and the disease has become progressive within the herds of buffaloes with 92% prevalence. At this rate, all herds of buffaloes will eventually become infected with bovine tuberculosis. Buffaloes are considered as maintenance hosts of bovine tuberculosis, and its movement has to be restricted to prevent the disease from spreading to other rare or endangered wild species. Buffaloes are highly valued animals in ecotourism and trophy hunting industries. Thus, they are high in demand, and the disease-free animals in auctions can fetch prices up to millions. Farmers and national parks are affected financially through revenue losses due to restriction in the movement and sale of buffaloes which are in place. The disease is also zoonotic, therefore human population can easily become infected if coming in close contact with an infected animal. Disease surveillance. Kruger National Park has an ongoing surveillance program to test for bovine tuberculosis prevalence. Survey is also ongoing in southern, central, and northern areas of the park, which shows high prevalence of disease, especially in the southern part of the park. Between 1991 and 1992, about 2,071 buffaloes have been culled through active surveillance. In 1998, a park-wide monitoring exercise examined about 618 buffaloes which were lethally sampled and examined for bovine tuberculosis, which showed a rise in the disease prevalence. In 2000, non-lethal blood-based gamma interferon test was done, which sampled about 608 buffaloes, where 5 showed positive signs of bovine tuberculosis. A buffalo TB research project has also started at the park since year 2000 to collect data on how ecology, epidemiology, and demographic processes influence the spread and impact of bovine tuberculosis in African buffaloes. Controlling bovine tuberculosis in Kruger National Park. The first method is through capture, test, and slaughter policy. Buffaloes are mass captured in Kruger National Park, and all animals that test positive are slaughtered, and animals which are tested negative are then released into the park. The second method is through an effective breeding program which has been established at Kruger National Park to produce disease-free buffalo calves, which are then removed from the park and restocked in the conservation areas. Thirdly, the use of vaccines. Bacillus calmette guerin, also known as BCG vaccine, is currently available, but they are doing a further research to examine its effectiveness on buffaloes against bovine tuberculosis. And lastly, the use of electric fence. Electric fence has been successfully installed surrounding the Kruger National Park to prevent buffaloes from coming in direct contact with domestic cattle, which are the primary host of bovine tuberculosis in South Africa. Ebola virus outbreak in West Africa in 2014. History of the disease. The disease was first discovered in Democratic Republic of Congo in 1976, where it caused 280 deaths, and in the same year the disease caused 151 deaths in Sudan. Further sporadic outbreaks have occurred in African countries such as Uganda in 2000 and in 2007, and in Congo in 1995, 2007, and 2012, before the major outbreak in West Africa in 2014. 2014 Ebola outbreak in West Africa. On 23rd of March 2014, World Health Organization, also known as WHO, was notified of Ebola outbreak in Guinea. On the 8th of August 2014, WHO declared Ebola epidemic and declared the disease as a public health emergency of international concern. Countries most affected were Guinea, Liberia, and Sierra Leone. The map on this slide shows the location of the three countries as they are bordering each other in West Africa. Epidemiology of Ebola. Firstly, its cause. Ebola is caused by virus from the family known as Filoviridae. It belongs to the genus known as Ebola-like virus, and the species is known as Ebola. The virus is endemic in Africa, especially in wildlife species. Host and disease transmission. The picture on this slide shows fruit bats act as the reservoir for Ebola virus and spread the virus to other wildlife species, including human population. Humans get infected with the virus when coming in contact with either the fruit bats or the wildlife species. And once infected, humans become contagious and then they spread the virus to other humans. How the disease spreads to humans. The slide shows how the disease spreads to human population from wildlife species. Humans get infected with the virus firstly when they eat fallen fruits that were partially eaten by fruit bats. Secondly, by eating infected fruit bat meat. And lastly, by eating or handling uncooked bush meat, in most cases, meat of monkeys. Human to human disease transmission. This slide shows how the virus spreads from one human to the others. Firstly, it is very important to know that humans only become contagious after showing the symptoms of Ebola. Methods of disease transmission includes when humans attend burial ceremonies and come in direct contact with the dead body. Also, the disease transmits through direct contact with blood or secretion, such as urine, feces, vomit, spit, sweat, semen, and breast milk of the infected person, or when humans are exposed to objects or environment contaminated with infected secretions. And lastly, when treating patients who are infected with Ebola virus. In most cases, the healthcare workers, such as doctors and nurses, are at high risk because they are exposed to the patients. Symptoms of Ebola virus. The picture on the slide shows the symptoms of Ebola virus. Firstly, the incubation period of the infection to appear is usually between 3 to 21 days, and on average, the symptoms would appear within 8 to 10 days. Ebola virus symptoms are described in three main categories. Firstly, the general symptoms, which appear within 3 days. The patients will have fever-like symptoms such as severe headache, muscle pain, sore throat, weakness, and tiredness. Secondly, gastrointestinal symptoms, which normally will appear within 3 to 10 days, and the patients will have vomiting, diarrhea, and abdominal pains. And lastly, severe Ebola symptoms, which generally will appear within 7 to 21 days. The patients will have severe diarrhea and hemorrhage, bleeding through nose and intestines, and the patients will have regular vomiting and may eventually die. Impact of Ebola virus. The next two slides will show the impact of Ebola virus in West Africa, especially in the three most worst affected countries. Firstly, the impact of Ebola virus on human population and health. It was huge. is about 17,300 children lost one or both of their parents due to Ebola virus and became orphans. Also, about 513 healthcare workers, including nurses, doctors, and midwives, lost their lives. In total, about 11,310 lives were lost, which included 2,544 lives in Guinea, 3,956 lives in Sierra Leone, and 4,810 lives were lost in Liberia. There was an increase in unemployment due to closure of commercial companies, and education sector was impacted as schools were closed during the outbreak. Travel and tourism. About 339,000 people were screened flying out of the most affected countries to prevent the Ebola virus from spreading. Movement restrictions were also in place for people affected with Ebola and general public, as borders were also closed off. International travelers were warned not to travel to Ebola affected countries, which impacted tourism industries in West Africa. And the neighboring countries to the three most worst affected countries also closed their borders to restrict the movement of people. Impact of Ebola virus on the economy. About $2.2 billion in GDP was lost in the three worst affected countries due to Ebola virus outbreak. For example, in Sierra Leone, about 50% of private sector lost half of its workforce, and agricultural sector was also affected as farmers stayed away from markets and their farmland. Cocoa industry, which is the main industry in West Africa, is about 70% of the world's cocoa comes from this area, was also threatened due to movement restrictions and trade bans during this outbreak. The response to control this outbreak cost well over $3.6 billion. Due to Ebola outbreak, potential investors were also reluctant to invest in the three worst affected countries. Therefore, these countries lost millions of dollars as well. Ebola virus surveillance. Ebola virus surveillance and monitoring was an important step to prevent the disease from spreading to other countries or from one infected human to another. Ebola response teams were assembled in each country to deal with the outbreak with the help of experts from international agencies. Some of the surveillance methods used are discussed in this slide. Firstly, surveillance through contact tracing, whereby people with Ebola symptoms or people exposed to the disease were identified, assessed, and managed to prevent the virus from spreading. This was the most important surveillance technique used. Secondly, alerts and hotline was also created to receive information on people with signs or symptoms of Ebola from the general public. Door-to-door knocking to actively find people which may have the Ebola symptoms was also practiced in remote areas. Travel restriction and passenger screening was done at the airports to assess the health of travelers coming into and going out of the country to prevent the disease from spreading. Social mobilization and community awareness was also effective. Community leaders and volunteers were used to create awareness about the disease, especially handling of dead bodies and use of proper protective equipment and following strict hygiene procedures were explained or demonstrated to the general public to prevent the disease from spreading. Case investigation and reporting, whereby every case which was brought to the attention of the authorities were attended to, recorded, and reported to the Ebola treatment team. Treatment and prevention of Ebola virus. The World Health Organization declared the end of Ebola transmission in Guinea on 29th December 2015, in Liberia on the 14th of January 2016, and in Sierra Leone on the 17th of March 2016. These countries entered 90 days of surveillance to ensure no new case of Ebola was identified in order to be declared Ebola free. This was achieved by the following methods. As it is known that at present there are no vaccines available to control the disease, therefore traditional methods such as early recognition and isolation, whereby patients with Ebola or showing the signs of the disease were identified through contact tracing and tested and isolated to prevent transmission of the virus, was the key step to combat the disease. Secondly, all medical staff were trained to handle Ebola infected patients and were to wear proper protective equipment when dealing with Ebola infected patients. Hygiene and sterilization was critical, as people were informed to follow simple hygiene procedures such as washing hands with soap and water or alcohol-based sanitizer, and to avoid contact with infected patient body fluids such as blood, urine, saliva, vomit, and semen, or coming in contact with contaminated medical equipment which were used on Ebola infected patient. Proper burial procedure was to be followed, and people were encouraged to avoid funeral or burial rituals that required handling someone who had died due to Ebola virus. People were also encouraged to avoid contact with wild animals, especially eating fallen fruits and meat of bats and monkeys. All incoming and outgoing passengers were screened by teams to prevent Ebola virus from spreading into the country or going out of the country. And lastly, all general public were also encouraged to wear proper protective equipment when dealing with members of their family who were potentially infected or were showing signs of Ebola virus. Finally, conclusion. Wildlife animals are equally important as domesticated animals, and human intervention plays a significant role in the development of infectious diseases in wildlife population. Therefore, research should also emphasize on the health and welfare of wildlife population and continue to develop medication and vaccines against diseases which are currently affecting their population. Public awareness is also important regarding deadly diseases which originates from wildlife population that are zoonotic. Therefore, research to develop vaccines against zoonotic diseases that originates from wildlife that have recently become a threat to human population, example Ebola, is necessary. Thank you.